The overall goal of this procedure is to identify and assess antibiotic susceptibility of blood culture isolates in less time than with conventional methods. This is accomplished by first identifying the blood culture isolate using a novel 16S ribosomal DNA multiprobe PCR assay. Then the isolate is incubated with a panel of antibiotics for 6 hours. The next step is to determine growth or growth inhibition of the isolate in the presence of the antibiotic using a quantitative 16S ribosomal DNA PCR assay. Ultimately, the results show that identification and antibiotic susceptibility testing of a blood culture isolate can be obtained more rapidly through this combination of culture and PCR.
The main advantage of this technique over existing methods, such as those using automated Phoenix or Vitex systems, is that no prior culturing step is required, and that results from identification and antibiotic susceptibility testing can be available within the same day. Demonstrating the procedure will be Wendy, Hansen and Judith Beuving, PhD students from our laboratory.
Dilute 100 microliters of blood culture positive for microbial growth in a reaction tube containing 900 microliters of 0.9% sodium chloride. Then, centrifuge the sample at 13,400 times G for 5 minutes to pellet the bacteria. Following the centrifugation, resuspend the pellet in 100 microliters of sterile demineralized water and store the sample at 4 degrees Celsius